Welcome back to Chapter 17, Part 3. In this video, we're focusing on Section 17.3, which looks at the cellular defenses of the innate immune system. We'll look at the different types of blood cells, where they come from, and how they protect the body from infection. By the end of this video, you should be able to identify and describe the components of blood, explain how the formed elements of blood are produced through hematopoiesis, and describe the major types of leukocytes, their appearance, and their functions within the innate immune system. Cellular defenses refer to the white blood cells, also known as leukocytes, that participate in innate immunity. And blood contains three main categories of formed elements. We have red blood cells, our white blood cells, those leukocytes, and platelets. So the cellular defenses focus on the innate immune white blood cells, which include granulocytes, agranulocytes, natural killer cells, and phagocytes. And all of these originate from hematopoietic stem cells, also abbreviated HSCs, from the bone marrow. Hematopoiesis is the process of producing all blood cells inside the red bone marrow. Multipotent hematopoietic stem cells, or HSCs, receive chemical signals that push them down different differentiation pathways, and there are two major lineages. Myeloid stem cells are going to produce red blood cells, platelets, granulocytes, and monocytes, whereas lymphoid stem cells produce natural killer cells, B cells, and T cells. And most hematopoietic stem cells become erythrocytes, those red blood cells, while proportionally fewer become leukocytes, those white blood cells. This diagram shows how every formed element comes from the same hematopoietic stem cell, and we can follow the branches to see that we get myeloid stem cells, which generate those platelets, red blood cells, and granulocytes, whereas the lymphoid stem cells generate those natural killer cells and the B and T cells. The function of red blood cells, or erythrocytes, is to help transport oxygen. Platelets help with clotting and tissue repair, and leukocytes, those white blood cells, provide immune defense. Leukocytes are divided into two main categories. We have our granulocytes, which have visible cytoplasmic granules and include the neutrophils, eosinophils, and basophils, and the agranulocytes, which lack visible granules and include monocytes, which will differentiate into macrophages and dendritic cells, as well as lymphocytes. This table from our book provides a good summary of each cell type, their appearance, and their functions, but we're going to zoom in so we can focus on each half of the table and see it a little bit more clearly. Here we have those red blood cells, the erythrocytes, and then the white blood cells, the leukocytes. Of the leukocytes, some of them are granulocytes, so we can see these include the neutrophils, eosinophils, and basophils. And if I zoom back out again, I can see red blood cells, white blood cells, and platelets. We just looked at the granulocytes, those three over here. We're going to zoom in next on those agranulocytes. And those agranulocytes include our lymphocytes and our monocytes. Within this category, we're going to focus on the B and T cells more in Chapter 18 for the adaptive specific immune system, but we will look at natural killer cells in this chapter. Let's look at the granulocytes first. Granulocytes are white blood cells that are identified by the number of lobes they have in their nucleus, as well as by how their granules stain. And we have those three types: neutrophils, eosinophils, and basophils. These all contain granules that contain potent enzymes and chemicals used to attack pathogens. And here's a closer look at our granulocytes. We have our neutrophils, eosinophils, and basophils. And it turns out that eosinophils stain really well with an acidic stain we learn about in lab called eosin. You can see the reddish granules here. The basophil cells stain really well with methylene blue. Methylene blue is a basic stain, and you can see the large granules there. They look kind of bluish purple because of that methylene blue. And neutrophils stain with both eosin and methylene blue. So this is acidic, this one is basic. So together we call them a neutrophil because these are staining with collectively neutral stains. Our first granulocyte are the neutrophils, and these are also called the polymorphonuclear neutrophils, or PMNs. These are the most abundant white blood cells, and they respond rapidly to bacterial infection and use three main killing mechanisms: phagocytosis, degranulation, which means they release toxic molecules to kill microbes. It just so happens that when many neutrophils are brought into an infected area, they can be stimulated to release these toxic molecules into the surrounding tissue, and that will help destroy those infectious agents or pathogens. And this process is called degranulation. Neutrophils can also form nets. Nets stands for neutrophil extracellular traps, and these are made of chromatin and antimicrobial chemicals that together trap and kill microbes. So neutrophils arrive to the infection site quickly, and their accumulation along with the cellular debris and bacteria is what forms pus during infections. Eosinophils are our next granulocyte, and these have bilobed nuclei, or 2 to 3 lobes in their nuclei, and distinctive red-orange granules. They specialize in fighting protozoa and helminths, which are too big for phagocytosis. And their granules contain major basic protein, or MBP, which damages parasite membranes. Eosinophils also contribute to allergic responses. Our third granulocytes are basophils, which have dark blue to purple granules that can obscure much of the nucleus. And they play a major role in inflammation and allergic reactions. Their granules contain histamine as well as other inflammatory mediators, and histamine can be chemotactic and attract other white blood cells to the area, and can also help open up the gaps between the cells and the blood vessels, so cells can leave the blood and enter the tissues that are damaged. Basophils can be activated by the complement proteins C3A and C5A, and remember these were previously called anaphylatoxins, and when this occurs, this triggers degranulation of the basophils. Mast cells are similar to basophils, but they differ in one key way. Mast cells reside in tissues, not in the bloodstream. So if we look down here, we see a few mast cells, and here they're indicated by the arrows found within tissues. So unlike basophils, mast cells migrate from the blood into various tissues. They're found in the skin, mucous membranes, and near blood vessels and nerves. They also release histamine and contribute to allergic responses as well as local inflammation. This first checkpoint asks, describe the granules and nuclei of neutrophils, eosinophils, basophils, and mast cells. So neutrophils have about 3 to 5 lobes, so we call them polymorphonuclear, and their granules are small and numerous. They're generally lilac-colored or neutral-colored granules that contain chemicals like defensins as well as hydrolytic enzymes. For eosinophils, they have about 2 to 3 lobes. Their granules are large, reddish-orange in color, and they contain histamine, degradative enzymes, and that major basic protein. For our basophils, they have 2 lobes. Their granules are large, dark blue and purple, that are rich in histamine and inflammatory mediators. And for mast cells, they have a round nucleus. It's not lobed. And their granules are similar to basophils. They're packed with histamine as well as other inflammatory mediators. But remember, mast cells are found in tissues, especially in the skin and mucosal surfaces. The second checkpoint asks, name 3 antimicrobial mechanisms of neutrophils. So we saw that neutrophils can kill microbes in different ways. One is through phagocytosis, where they engulf and digest the pathogen. Second was degranulation, where they release toxic molecules into the environment that target those microbes. And the third was those nets, the neutrophil extracellular traps that are made of these matrices of chromatin and antimicrobial proteins that can bind to, capture, and kill the pathogens. So we just covered the granulocytes. We looked at the neutrophils, eosinophils, and basophils, and we also looked at mast cells. These are not granulocytes, but they are kind of like or similar to granulocytes because they have histamine and behave similarly to basophils. Now we're going to move on and look at the agranulocytes. More specifically, we'll look at monocytes, natural killer cells, as well as a brief intro to those lymphocytes. Our agranulocytes lack visible cytoplasmic granules, and these include the lymphocytes, including the natural killer cells, B cells, and T cells, as well as the monocytes, which are going to further differentiate into macrophages and dendritic cells. And we're going to see that these cells play key roles in both innate and adaptive immunity. Let's start with natural killer cells. Natural killer cells, or NK cells, are lymphocytes that kill virus-infected or cancer cells using non-specific mechanisms, and they're activated under two conditions. The first is when MHC1 decreases. MHC1 stands for Major Histocompatibility Complex 1, and this marks the surface of healthy cells. So in our normal healthy cells, on the surface of those cells, we have a marker called MHC1. And we're going to talk more about these in Chapter 18. But if a cell has been infected by a virus or has become cancerous, then it's no longer healthy, and you're going to see that the number of MHC molecules on the surface of these cells goes down. The second mechanism or condition is activating molecules. So when cells are unhealthy because they are infected by a virus or have become cancerous, they're going to have greater numbers of activating molecules on their surface, and that indicates that the cell has been altered or is unhealthy. So natural killer cells will kill these cells, kill these virus-infected or cancer cells, these are their targets, by either using perforin or granzyme, or a combination of the two. Perforin forms pores on the surface of the infected or cancerous cell, and water will rush in and lyse those cells. Granzymes can also enter those cells to trigger apoptosis or cell suicide. This is also known as programmed cell death. And this will prevent the cancer cells from continuing to divide or prevent the virus from spreading. On the right, we can see these are natural killer cells with a bunch of perforin-containing granules within them. Here we can see natural killer cells. In the first scenario, we are looking at a healthy cell. Here is the natural killer cell. The healthy cell is expressing Major Histocompatibility Complex 1 on its surface, indicating that it's healthy. So if a natural killer cell binds to that MHC1 complex, it realizes the cell is healthy and does not kill it. Nothing happens to that cell. But if it encounters a cancer cell or a virus-infected cell, there will be reduced expression of MHC1 in addition to an increased expression of those activating molecules. And when that happens, for instance, in this infected cell that's been infected by a virus, you can see there's no MHC1 on its surface, but there are activating, or at least there's one activating molecule shown. That triggers the natural killer cell to release those perforins and granzymes to create pores in the cell, as well as trigger apoptosis or cell death. Our next agranulocyte are monocytes, and these are large agranular white blood cells with a kidney-shaped nucleus. They circulate briefly, but then they migrate to tissues or into tissues. And once they get into the tissues, they differentiate into macrophages and dendritic cells. And their main roles are phagocytosis and cytokine production. Remember, these are cell communication molecules. So here are our monocytes, which can differentiate into macrophages, and macrophages are long-lived tissue phagocytes, and they vary widely depending on which tissue they're found in. Examples include microglia in the brain, the Kupffer cells in the liver, and the alveolar macrophages in the lungs. These all engulf pathogens. They can also engulf debris. They can produce those cell signaling molecules, those cytokines, and they will also link the innate and adaptive immune systems by presenting antigens. And we'll talk more about this in chapter 18. For the dendritic cells, these are found in the skin and mucous membranes, the places where pathogens are most likely to enter. They are highly phagocytic and also produce strong cytokine responses. And their most important function is antigen presentation. This is going to activate T cells and initiate adaptive immunity, which we'll talk more about in chapter 18. So because these are going to be encountering pathogens at the most likely entry points through those skin and mucous membranes, we call them our major sentinel cells. This table from our book lists the macrophages by their tissue-specific names. We have microglia in the CNS. We have the Kupffer cells in the liver, alveolar macrophages in the lungs, and I didn't have a picture of the peritoneal macrophages, but these are found in the abdominal cavity. These cells share very similar functions, but they're specialized for their tissue environments. All right, so a quick overview. We had our granulocytes, which included the neutrophils, eosinophils, and basophils. And we also included our mast cells in this discussion because although they're not granulocytes, they're granulocyte-like and behave very similarly to basophils. For our agranulocytes, we saw our natural killer cells that kill abnormal cancer or virus-infected cells non-specifically. We also had our monocytes that differentiate into macrophages and dendritic cells. And these also include our lymphocytes, but these are more a topic for chapter 18. To summarize some of the key ideas from this section, we saw that all white blood cells originate from hematopoietic stem cells in the bone marrow. Granulocytes are fast responders that use granule-mediated killing. Natural killer cells killed self-altered or altered self cells. So these are abnormal cells that are no longer healthy. Our monocytes, macrophages, and dendritic cells are the major phagocytes of immunity, and dendritic cells are going to be the bridge between the innate and adaptive immune responses. This first checkpoint asks, describe the signals that activate natural killer cells. So natural killer cells become activated when inhibitory self signals are missing. So they have reduced or absent MHC1 on the target cells that are seen in virus-infected or cancer cells. So the loss of the MHC removes that don't kill me signal. Or we can have activating signals be increased. So target cells display activating molecules such as altered self markers, and when these activating signals go up and the inhibitory signals, those MHC1 goes down, then the natural killer cell will kill the abnormal cell. For the second question, what is the difference between monocytes and macrophages? Monocytes circulate in the blood. These are large agranular white blood cells, and they're pretty short-lived in circulation. They have not yet fully differentiated. Whereas for macrophages, they form when the monocytes have migrated into tissues, and these are fully differentiated tissue-resident phagocytes. They are long-lived, highly phagocytic, they make cytokines, and they support tissue function. They also have tissue-specific names like those Kupffer cells, alveolar macrophages, and microglia. All right, let's recap 17.3. The first learning objective asks, identify and describe the components of blood. So blood has two major components. We have the plasma, or the fluid portion, which is mostly water, proteins, electrolytes, nutrients, and chemical mediators like complement proteins and the acute phase proteins that we saw in section 17.2. We also have formed elements, the cellular portion that we saw in this section, including our red blood cells, those are also known as erythrocytes, our white blood cells, also known as leukocytes, and those included the granulocytes and the agranulocytes. And we also have platelets that are involved in clot formation. For the second learning objective, explain the process by which the formed elements of blood are formed, or hematopoiesis. Hematopoiesis is the formation of all blood cells from our stem cells, starting with the multipotent hematopoietic stem cells. Those were found in the bone marrow. And from those cells, we had the myeloid lineage, and the myeloid lineage produces erythrocytes, platelets, granulocytes, and monocytes, whereas the lymphoid lineage produces the lymphocytes, those T cells, B cells, and natural killer cells. Remember, from the hematopoietic stem cells, most of them become red blood cells, fewer become white blood cells. All right, for the third learning objective, describe the characteristics of formed elements found in peripheral blood, as well as their respective functions within the innate immune system. So red blood cells, they don't have a nucleus, they carry oxygen, they're not part of the innate immune system. Platelets, they're involved in clot formation and wound repair. They can help trap microbes within the clots. It's really the white blood cells or leukocytes that come into play. We had those granulocytes. The neutrophils can go through phagocytosis, degranulation, and form those nets. They are the main responders to bacterial infections. We also had the eosinophils that attack helminths and protozoa, and the basophils that are involved in inflammation and allergic responses and release histamine. For the agranulocytes, we saw the natural killer cells kill virus-infected or cancer cells. We also had our monocytes. Our monocytes differentiate into macrophages or dendritic cells. Those macrophages, we saw them in tissue-specific regions with specific names like microglia or alveolar macrophages, and they're primarily involved in phagocytosis and cytokine signaling. For the dendritic cells, we saw them mainly in the skin and mucous membranes. They're also strongly phagocytic, and we're going to see that they will eventually present antigens that link innate immunity to the adaptive immunity we'll talk about more in the next chapter. And that wraps up section 17.3 on cellular defenses. In the next video, we're going to move on to 17.4, pathogen recognition and phagocytosis.